welcome back to my channel. Today we'll speak about nucleotides, both DNA and RNA. But first we're going to start with nucleic acids. There are two chemically distinct types, which is DNA and RNA. Now, they are required for the storage and the expression of genetic information. The bases, so there are 4 types of bases in DNA and 4 in RNA, but there is only one difference. So the bases are thymine, adenine, cytosine, guanine, and uracil. So thymine, adenine, cytosine, and guanine are in DNA, and the same are in RNA except that the thymine is replaced with uracil. The bonding is such that it is called complementary base pairing, that adenine only binds with thymine, and there are 3 bonds between them. And in RNA, adenine will bind with uracil, 3 bonds. Cytosine and guanine only bind with one another, and they have 2 bonds between them. The structure of nucleic acids is that they are linked by phosphodiester bonds. Now, these can be cleaved by enzymes of family nucleases. So deoxyribonucleases for DNA, because DNA stands for deoxyribonucleic acid, and ribonucleases for RNA, which is ribonucleic acid. Okay, let's start with DNA. A few viruses contain single-stranded DNA, but it is mostly double-stranded in humans and in bacteria, in which the 2 strands wind around each other, forming a double helix. There are 3 major structural forms of DNA. There is the B form, the beta, the A form, alpha, and the Z form. The most common one is the beta form, which is a right-handed helix with 10 residues per 10 of the helix. There is the semi-conservative replication, which means that each strand of the double helix, it serves as a template for constructing a complementary daughter strand. Now, the replication is bidirectional. Helicase is the enzyme which unwinds the double helix. DNA topoisomerases types 1 and 2 are 2 other enzymes which remove the supercoils from the DNA. Then there's another enzyme, the DNA polymerases. They synthesize new strands only in the 5' to 3' direction. Now, DNA polymerases, they require a primer, or like a template again, which is a short stretch of RNA synthesized by primase. The function of the DNA is that it contains the hereditary information, and it is important for the future generations. The leading strand goes 5' to the 3' and does not require the enzyme ligase because it is continuous. But the lagging strand needs ligase enzyme because it is discontinuous. So it keeps having small breaks and intervals and starts forming the new DNA with little fragments. Now, moving on to RNA, there are 3 major types: rRNA, tRNA, and mRNA. It is smaller than DNA and contains ribose instead of deoxyribose, which is the sugar present. They exist as single strands. So let's look at each of them individually. rRNA is ribosomal. RNA is the component of the ribosomes, which are the sites of protein synthesis. Then you have tRNA, which is the smallest of the 3 RNAs. They are specific for each of the 20 amino acids, and they carry the amino acid covalently attached to its 3' end, which is the site of protein synthesis. So they take the amino acids to the ribosomes. And then you have the mRNA, which is the messenger RNA. It carries genetic information from the nuclear DNA to the cytosol, where it is used as a template for the protein synthesis. The enzyme that synthesizes RNA is known as RNA polymerase. The process through which the protein is, the mRNA is made in the nucleus of the cell is called transcription. It is made from the DNA template, and the process where the protein is made in the cytosol from the ribosomes is called translation. So that is it for this video. Hope you guys enjoyed it. See you in the next one. Bye.